Imagine a blood test that tells you a lung tumor will form in 5 years. But, more importantly, it gives you the exact blueprint to stop it today.
Right, which is just incredible.
Yeah, it is. Because we aren't really talking about early detection anymore. We're talking about detecting the actual soil that allows cancer to grow long before the seed even sprouts.
Exactly. It's a complete paradigm shift in how we approach medicine. I mean, historically, we just wait for a physical manifestation of disease, right?
Shadow on an X-ray.
Right, a shadow, a lump. But biology doesn't just happen overnight. There is this silent cascade of cellular events that takes years, and we are finally, learning how to listen to that specific conversation.
And that's what we are unpacking today. We're looking at this massively comprehensive paper published in the journal Cell.
A really landmark paper.
Totally. So a global team of researchers discovered a 14-protein signature in the blood that acts as a highly precise early warning system for lung adenocarcinoma. And our mission today is to tear this entire process apart.
Get right into the weeds.
Yeah, we want to know how they found these proteins using machine learning,
Right.
Where exactly in the lung tissue they originate, how everyday air pollution, sparks the fire, and finally, how we can clinically intercept this process to prevent the cancer from ever forming. Okay, let's unpack this.
My philosophy here is that knowledge is most powerful when we understand the mechanisms behind it. If we can understand the specific environment, the stress, damaged state of the lung, we stop reacting to cancer and start dismantling the infrastructure it needs to survive.
So let's trace this from the very beginning, because to find a pattern, you need a staggering amount of data.
Oh, absolutely.
The researchers utilized the UK Biobank, which is basically a gold mine for this kind of longitudinal study. They started with 48,099 people.
That's a huge cohort.
Right. And they took baseline plasma samples from all of them and measured nearly 3,000 different proteins, specifically 2,923 proteins. Then they simply waited for half a decade. Over an average of 5.6 years, 375 of those individuals developed lung cancer.
Which, from a data perspective, presents a monumental problem.
How so?
Well, you have thousands of variables, right, and only a few hundred positive outcomes. If a human tries to find the correlation between roughly 3,000 proteins and a cancer diagnosis 5 years later, the math is impossible.
Yeah, our brains just can't do that.
Exactly. So they deployed a machine learning architecture called XGBoost, which stands for extreme gradient boosting.
Okay, I hear terms like XGBoost thrown around a lot in tech circles.
Yeah.
But how does it actually work in a biological context? How does it find a biological rule instead of just finding random statistical noise?
It works by building a series of decision trees. So imagine a flowchart that asks a question like, is protein A high or low? Depending on the answer, it moves to another protein.
Okay, makes sense.
XGBoost builds thousands of these trees, and the boosting part means that every time it builds a new tree, it specifically focuses on the cases that the previous tree got wrong.
Oh, so it learns from its mistakes iteratively.
Right, zeroes in on its errors.
Mm-hmm.
Now, to ensure it wasn't just, memorizing this specific UK Biobank data set, a problem data scientists call overfitting, they trained the algorithm on 75% of the data.
And then the rest.
They kept the remaining 25% locked away in a vault. They only tested the final model on that hidden 25% to prove it had actually learned universal biological rules.
But they didn't keep all 3,000 proteins, right?
Mm-hmm.
They used a process called recursive feature elimination.
Yes, to narrow it down to just 14.
Let's visualize how that works for the listener. Imagine you have an orchestra of 3,000 instruments. If they all play at once, it's just a wall of static.
Right, total chaos.
So the algorithm essentially mutes the instruments that are just playing random noise. Then it mutes the instruments that are playing the exact same melody as someone else because, they're redundant.
Exactly.
It systematically eliminates the least essential players over and over until it is left with a tight, perfectly synchronized 14-piece band that is playing the specific pre-cancer tune.
That synchronized tune is the absolute key here, because these 14 proteins aren't random markers. They represent a state of profound distress in the lung tissue.
To make this tangible, let's group these 14 proteins into 4 biological mechanisms. We can use the acronym MISS to track what this distress signal is actually doing. MISS.
That's a great way to remember it. So the M is for matrix remodeling.
What does that mean in the lung?
Well, the extracellular matrix is the structural scaffolding of your lung. One of the key proteins here is an enzyme called MMP12. A tumor cannot grow in a confined space. It needs physical room.
It needs a clear path.
Exactly. So the damaged tissue releases MMP12, which literally digests and breaks down the lung scaffolding to make that room.
Wow. Okay, then we have the I, which stands for inflammation. These are the chemical alarm bells, right?
Yeah, things like CXCL17 and GDF15. When tissue is damaged, it releases proteins that scream for help, pulling immune cells into the area.
Got it. And the first S is secretion and shedding. When the cells lining your airways are under immense stress, their internal machinery starts to break down.
Right, and they shed abnormal stress proteins like CECAM5 directly into the bloodstream.
And the final S is surfactant production.
Yeah, so surfactant is that slippery liquid that coats the inside of your lungs so they can expand and contract smoothly.
Like oil in an engine.
Perfect analogy. In a precancerous state, the cells responsible for making this fluid go into absolute overdrive, trying to protect the delicate air sacs. Proteins like LAMP3 and SFTPD spike.
So we have matrix remodeling, inflammation, secretion, and surfactant, MISS.
Exactly.
And the researchers proved this wasn't just a quirk of the UK population. They validated this exact 14-protein signature across 8 different global cohorts.
Which is incredibly robust.
Yeah, and it drastically outperformed the standard clinical risk models doctors use today, especially in that hidden window 2 to 4 years before a tumor is even visible on a scan.
It sees what a CT scan can't.
But there's a detail in the data that completely threw me. They looked at a group called the TRACERx cohort. These were patients who already had lung cancer, and surgeons physically cut the tumors out of their lungs.
Right.
I would assume that if you remove the tumor, this 14-protein alarm bell in the blood would go silent. But it didn't. Why on earth would the signature stay high if the cancer is sitting in a surgical tray?
Because the tumor was never the source of the alarm.
Wait, really?
Yes. The 14 proteins are being pumped out by the surrounding lung tissue. The tumor is just the eventual consequence of a sick environment.
Oh man, so removing the tumor is like picking a weed.
Exactly. If the soil is still toxic, the chemical signature of that toxic soil remains completely unchanged.
That is wild. It means the blood test is measuring the soil, not the weed. And this isn't just about heavy smokers ruining their lung tissue either.
No, not at all.
They tested this in the talent trial in Taiwan, which focused entirely on people who had never smoked a cigarette in their lives.
A very important demographic.
And even in those never smokers, this exact same MISS protein signature spiked years before they were diagnosed with a specific genetically driven lung cancer.
Which reveals a unified biological response. Whether the lung is damaged by a lifetime of heavy smoking or driven toward cancer by a spontaneous genetic mutation in a non-smoker, the tissue reacts the exact same way. The distress signal is universal.
Okay, let's do a quick recap. We've used machine learning, XGBoost, on UK Biobank data to filter thousands of variables down to a 14-protein distress signal. We categorized them by MISS, and we proved this signal is coming from the soil of the lung, predicting cancer 5+ years out.
A really solid foundation.
So, listener, think about your own environment for a second. If your body's check engine light came on 5 years early, what would you do? What stands out to you about those MISS categories? Because we need to figure out where exactly this is happening.
Right, the lung is a big place.
Exactly. So where in the lungs are these proteins actually being manufactured?
To answer that, the researchers had to go down to the cellular level using some incredibly precise genetic mouse models. You really have to understand the architecture of the lung first.
Okay, lay it out for us.
When you breathe in, the air travels down a series of branching tubes, and the cells lining these tubes change depending on how deep you go.
Let's use a mental map for you guys listening. Picture the lung as an upside-down tree. The sturdy trunk is made of basal cells. The branches are lined with club cells.
And at the very ends of the branches, you have the fragile leaves.
Right, the tiny air sacs called alveoli, where oxygen actually enters your blood. These leaves are made of alveolar type 2 cells, or AT2 cells.
The researchers wanted to know which of these specific cells was capable of starting the cancer, so they used inhaled viruses to selectively deliver specific genetic mutations to the mice.
How do they track that?
They created different groups. Some mice just received a fluorescent red tracking gene called a TD tomato reporter, so their cells would physically glow under a microscope.
Okay, that's group 1.
Others received the tracker, plus a dangerous mutation called EGFR, which is the exact mutation driving the spike in cancer among non-smokers we talked about. A third group received the tracker, the EGFR mutation, and the removal of a critical gene called TRP53.
TRP53, that's a tumor suppressor, right? Basically the brakes of the cell.
Exactly. Without it, the cell is highly prone to multiplying out of control. Now, by using those targeted viruses, they could drop these mutations exclusively into the basal trunk cells or exclusively into the branch cells.
And the physical behavior of these cells is what blew my mind. No matter where they injected the mutation, even if they mutated the sturdy basal cells in the trunk, those specific mutant cells detached from their neighbors.
Yes, they became mobile.
They physically migrated all the way down into the fragile alveolar leaves. They packed their bags and moved to a new neighborhood before they started forming a tumor.
It's an incredible migration to watch on a cellular level.
But hold on, if these mutant cells are physically traveling through the lung tissue, why doesn't the body's immune system flag them as rogue and destroy them in transit?
Because they are hiding in plain sight. They aren't behaving like cancer cells yet. They are behaving like repair cells responding to damage.
Sneevy.
Very. And this brings us to a crucial biological mechanism called the KAC state. KAC stands for keratin 8 positive, claudin 4 positive alveolar transitional state.
That is a mouthful. Let's unpack that.
Let me break it down. In a healthy lung, if those delicate alveolar leaves get damaged, the local AT2 cells act as what we call facultative progenitors.
Wait, facultative progenitors, meaning they have the option, the faculty, to act like stem cells if there's an emergency.
Precisely. They are normal working cells, but under stress, they change their identity. They morph into a temporary repair state to patch the holes in the lung.
Okay, that's normal biology.
Right. But when a cell has that EGFR mutation we just talked about, a catastrophic glitch occurs. The cell receives the signal to repair the damage. It starts changing its identity, but the mutation prevents it from finishing the job.
Gets stuck halfway through the repair process.
Yes.
It's like a caterpillar that spins a chrysalis, starts turning into a butterfly, but gets permanently frozen halfway through the transformation.
That is exactly it. And that frozen intermediate phase is the KAC state. It is a highly plastic, vulnerable biological bottleneck.
And the researchers proved this convergence using a technique called single-nucleus RNA sequencing, or cRNA-seq, right?
It did.
I want to spend a second on cRNA-seq, because sequencing usually makes people think of reading DNA. But DNA is just the master library of the cell. It holds every possible instruction.
It's a very important distinction.
RNA is the specific book the cell has checked out and is actively reading at that exact moment. By sequencing the RNA of individual single cells, the researchers weren't looking at what the cell could do. They were reading its active blueprints to see what it was currently building.
And when they cracked open these individual cells and read their active RNA blueprints, they saw something remarkable.
What was it?
Whether the cell originated in the basal trunk, the club branches, or the alveolar leaves, once it had the EGFR mutation, its RNA blueprint completely changed. Every single one of them abandoned their original identity and adopted the exact same KAC transcriptional program.
What's fascinating here is that multiple different starting points lead to the exact same biological bottleneck. All roads lead to Rome, or, the KAC state.
Exactly.
And here is the punchline of this entire cellular biology journey. When those cells are trapped in that frozen KAC state, what do? They start mass-producing R14 MISS proteins.
That is the source.
The distress signal isn't coming from a tumor. It is coming from thousands of microscopic cells that are permanently stuck trying to heal the lung.
It's a tragic irony, really.
So let's recap again. 4 lung cell types are given an EGFR mutation. They migrate to the alveoli. They get stuck in the KAC intermediate state. And this state pumps out the 14 protein signature.
Perfect summary.
So to you listening, if all roads lead to Rome, or in this case, the KAC state, could we just put a roadblock right there?
Yep.
We have a lung full of these stuck, vulnerable cells just sitting there like dried tinder. But they aren't cancer yet. What is the actual spark that ignites them?
The spark is our environment, specifically microscopic pollution known as particulate matter, or PM2.5.
So dirty air.
Yes, the invisible soot from car exhaust, wildfires, industrial emissions, and of course, cigarette smoke. When you inhale PM2.5, it travels all the way down into those delicate alveolar leaves.
And your immune system responds to that invasion naturally.
Yeah.
You have resident immune cells patrolling your lungs called macrophages.
Which are essentially cellular garbage collectors.
Right. Their job is to swallow foreign debris and protect the tissue.
But here's the problem. A macrophage cannot digest rock, heavy metals, or synthetic soot.
It's not food.
Exactly. It swallows the PM2.5, but the particles physically damage the macrophage from the inside out. The immune cell becomes highly debilitated and frustrated.
And what does a frustrated immune cell do?
It triggers a massive inflammatory alarm. It releases a highly potent signaling molecule called interleukin-1 beta, or IL-1.
Here's where it gets really interesting. IL-1 is the spark. We have those mutated cells stuck in the KAC bottleneck. When the macrophage releases IL-1, it washes over those stuck cells and acts like pure, high-octane fertilizer.
That's a great way to put it.
It chemically forces those KAC cells to start multiplying rapidly, pushing them out of their frozen state and directly into aggressive lung adenocarcinoma.
Right, so the environmental pollution doesn't necessarily cause the initial genetic mutation. The mutation might already be there, just quietly dormant.
Oh, wow.
Yeah, the pollution causes the specific macrophage inflammation, the IL-1, that feeds the mutation and forces it to grow.
Which fundamentally changes the narrative of cancer prevention. If the sequence is pollution causes macrophages to release IL-1, and IL-1 fuels KAC cells to become cancer, what if we just block the IL-1?
And that brings us to the clinical reality and a massive piece of level 1 evidence called the CANTUS trial.
Now, the CANTUS trial wasn't even about lung cancer originally, was it?
No, it wasn't. They were testing a drug called canakinumab for heart disease.
What is canakinumab exactly?
It's an anti-IL-1 monoclonal antibody. You can think of it as a highly engineered sponge that circulates in your blood, specifically seeking out and neutralizing IL-1.
So if the macrophage releases the inflammatory alarm, canakinumab absorbs it before it can reach the surrounding tissue.
Exactly. And during this heart disease trial, the researchers noticed a stunning side effect. The patients taking canakinumab had drastically lower rates of lung cancer.
It was literally preventing the tumors from forming.
Yes. But when oncologists subsequently rushed to give this drug to patients who already had established late-stage lung cancer, it failed completely.
Wait, why? If it stops lung cancer, why did it fail?
It failed because of the timeline we've been discussing. Canakinumab is not a cancer killer. It does not destroy tumors. It is a KAC cell fertilizer blocker.
Oh, I see.
It only works if you deploy it while the cells are still in that stuck precancerous state. Once the tumor has formed and established its own blood supply, blocking IL-1 is like turning off the sprinkler system during a Category 5 hurricane. It's just too late.
So what does this all mean for a patient today? The obvious solution would be to give canakinumab to everyone as a preventative measure, right? Treat it like a daily vitamin for people living in polluted cities.
You can't do that, and it comes down to a fundamental medical economics concept called the number needed to treat, or NNT.
Break that down for us.
NNT tells you how many people you have to give a drug to in order to prevent one single bad outcome. If you give canakinumab to an unselected general population, the NNT to prevent one case of lung cancer is over 140.
Meaning 139 people are taking a drug that does absolutely nothing for them.
Correct. And canakinumab isn't a benign vitamin. It suppresses a specific part of your immune system.
Right. Exposing 139 people to potential side effects like severe infections just to save one person from lung cancer is biologically reckless and economically impossible.
But this is where the 14-protein machine learning signature closes the loop. The researchers went back into the blood samples from the CANTUS trial. They wanted to see what happens if you only give the drug to people whose blood is actively screaming the MISS distress signal.
Oh, this is brilliant. So for the patients who had a high 14-protein signature, meaning their macrophages were actively choking on pollution and pumping out IL-1, and their KAC cells were in distress, what happened?
The number needed to treat dropped from over 140 down to just 55.
Wow. That drop changes the entire risk-benefit analysis of modern medicine.
Right.
You are no longer guessing who might get cancer based on their age or smoking history.
Exactly. You are biochemically proving who has the exact inflammatory fire burning in their lungs, and you are extinguishing it.
And the data for the other side of the coin was equally definitive, right? For patients with a low 14-protein signature, taking the drug gave them the exact same odds of getting lung cancer as taking a placebo sugar pill.
The drug does literally nothing for them. The odds ratio was 0.91 with a p-value of 0.803, which is statistically insignificant. If you don't have the specific IL-1 storm brewing in your tissue, blocking it provides zero benefit.
Time for a final quick recap. PM2.5 pollution enters the lungs. Macrophages eat it and release IL-1. That IL-1 fuels the stuck KAC cells. Canakinumab blocks the IL-1, and the 14-protein signature tells us exactly who actually needs canakinumab.
That's the entire pathway, from environment to intervention.
We've just gone from a complex machine learning algorithm to a targeted, highly specific cancer prevention plan. Listener, are you starting to see how everything connects?
If we connect this to the bigger picture, we are looking at the future of molecular cancer prevention. We aren't just waiting for tumors to form, we are measuring the inflammatory storm in the blood and neutralizing it. The synthesis of these disparate fields is what makes this research so landmark. We started with biobank data, used AI to isolate proteins, traced them to a cellular bottleneck, found the pollution trigger, and proved an existing drug can intercept it.
We are looking at a future where a routine blood draw tells your doctor that your lung tissue is stuck in a dangerous repair loop due to the air quality in your city, and they give you a targeted antibody before a single cancer cell actually forms.
It's incredible.
But as we wrap up this deep dive, I want to leave you with a thought that extends far beyond oncology. We've focused entirely on lung cancer today. But if you look closely at the supplementary data in this cell paper, this 14 protein signature didn't just spike in future lung cancer patients.
No, it didn't.
It also reliably spiked in patients who went on to develop COPD, chronic obstructive pulmonary disease, and IPF, idiopathic pulmonary fibrosis.
Which forces us to ask a profound physiological question. Are lung cancer, COPD, and IPF truly distinct diseases at their origin? Or are they simply different outward symptoms of the exact same underlying environmental injury?
If this 14-protein alarm bell is fundamentally measuring a damaged, stuck lung environment screaming for help, could targeting IL-1 be the key to stopping a whole spectrum of age-related respiratory diseases? Is lung cancer just one symptom of a deeper environmental injury?
That is the frontier we're looking at right now.
We are finally learning to treat the soil rather than just pulling the weeds. If you live in a dense city or near a wildfire zone, your immune system is likely negotiating with this particulate matter right now. It completely reframes how we should think about the air we breathe and the silent biological conversations happening inside us every second of the day.
It certainly makes you think twice about checking the air quality index.
Definitely. Keep questioning the world around you, keep taking your own deep dives into your environmental exposures, and we will catch you on the next deep dive.